I just gave a big introduction that I forgot to turn on the recording, so I'm not going to repeat it. Just about why this is important. You're going to hear that from our speakers. So thanks everybody for joining. So Denice Wardrop served as executive director of the Chesapeake Research Consortium, and she is a research professor of geography and ecology at Penn State University. Her discovery areas are wetlands of all kinds, landscape ecology, restoration of aquatic systems, adaptive decision-making, and how human activities impact the ability of natural systems to provide ecosystem services. She has served on science committees that advise both the Chesapeake Bay and Everglades restoration efforts, and passionately supports humans and aquatic systems finding ways to bring out the best in each other. In addition to her current role with CRC, she is a past chair of STAC and is one of the 2 lead editors on this forthcoming STAC report, which you're going to learn all about today, Achieving Water Quality Goals in the Chesapeake Bay, Comprehensive Evaluation of System Response. One other word, Denice took over in as CRC executive director, I think it was in like January of 2020, is that right? And her predecessor, Bill Ball, was actually secretary when this effort got started also. So I'm not sure whether he's going to make it here, but I want to recognize his efforts as well. It's been great to work with Denice and with Kurt and all these people. To me, this is like a family reunion. So I'm really happy that they're here. Denice, please take it away.
You're going to give it to Brian.
I know. Well, that's right. Yes, I'm sorry. Brian, you and Zach have the floor to say something about Kurt before Denice takes over. So let me put you guys on the screen. Okay, there's Brian, and I need to put Zach on the screen. Bear with me just a second. All right, you're both there. So please go ahead.
Well, I guess I'll start. Thanks, Andy. This is Brian at Virginia Tech. And like Andy said, with any family reunion, this is the awkward part of the family reunion, me starting off here. So, and the other thing is, I'm a firm believer is that you have to have a sense of humor, especially with things like this that involve so many people and take so much time. And so Andy, as I understand it, Andy wanted me to tell the backstory about Caesar, and so this is my tongue-in-cheek attempt to do that very quickly. So here we go. Hopefully it lands. So as you know, STAC is the Scientific and Technical Advisory Committee made up of many volunteers that rotate on and off, and have been very fortunate to work with those folks. And STAC meets quarterly in Annapolis. That's an important fact in this deal. So Zach Easton, who's on the line here, and he can retort at any time if he likes, but Zach and a colleague of Kurt Stephenson and mine at Virginia Tech, Zach never wanted to go to STAC the night before. Remember, we met quarterly. And depending on who's driving, Zach typically drove, but depending on who's driving, it takes about 5 hours to get from Blacksburg to Annapolis. So every morning when they would start off to come to the meeting, I would always had to be there the night before. Zach would drive, and most often Kurt would sleep for a while. But because, again, the STAC meetings opened up at 9 o'clock in the morning, so to get there in time, these guys would leave Blacksburg about 4 a.m. So as I understand it, Kurt would sleep a while on the road trips, but after he woke up, he would bend Zach's ear about various things, mostly Bay stuff. And then after the STAC meetings on the ride home, Kurt would again typically bend Zach's ear. And then occasionally they'd call me on my car as I drove home to Blacksburg as well. And Kurt would bend my ear and Zach's ear as well, as we talked about Bay stuff. And Zach, thankfully, was most often the grounding voice of reason in those miles-long conversations. And that's it. That's where Caesar came from. It was Kurt and Zach's intellectual curiosity and the dedicated road time. Eventually, through a variety of other settings, generally fires at one of our houses during the pandemic when we were distancing, we eventually convinced ourselves that Caesar was a good idea, and we presented this to STAC. And given STAC its charge to provide unbiased science-based advice to the Bay program, we thought that STAC was uniquely suited to pursue the Caesar effort. And STAC bought in. And so of the folks on the line here today, Dr. Miller and I are sort of co-conspirators or collaborators in that we helped steer STAC's involvement in this for a while. Dr. Wardrop was thankfully always the cool-headed, capable co-conspirator who helped us along. And the credit for Caesar then basically goes to Kurt and Zach. But it also, like Andy said, many others contributed and edited and reviewed the report, reviewed the report, helped us steer us in the direction we needed to go. And it's turned out to be a very, I think it's always been interesting. Sometimes it's been fun, but I think eventually it will be very impactful. So that's my story, Andy.
Thanks, Brian. I'm going to turn it over to Denice. I just need to do a little bit of bookkeeping on the screen setup here, so bear with me just a second. I need to make, well, you know what? I'm going to just take you guys off the stage, and then we'll let Denice take it. Apologize for the.
Just as a note, that's how many good ideas and things get hatched are in long car rides.
Sorry, just trying to clean this up. Okay, so Denice, I'm going to go ahead. We can share your slides.
Just for the record, Zach never let me drive.
All right, go for it, Denice.
All right, so as Brian implied, I'm just going to start off, and Kurt and I have tag team this presentation a lot, and every time we try to do something new, so we've switched up the order a little bit. So I just want you to know, especially being students, what's an advisory committee and why is this important that an advisory committee did that? STAC, of which Andy was chair, and Brian and Kathy Boomer is also online. It's a very unique group of people, and the advisory committee actually works on this thing. You'll see a check where it says STAC. The Bay program, when you have a partnership restoration effort, and if you didn't hear this in a previous seminar from Candace Boyd, what is always stunning to me is the partnership part of the Chesapeake Bay restoration. So just to clue you in, it's a 64,000 square mile watershed. It's the highest land to water ratio of any estuary in the world. That means what happens over those 64,000 square miles matters a lot. That takes a lot of people. It's 5 states and the District of Columbia, right? So it's a lot more complicated to get people to do something than I do a lot of work in the Everglades. It's much simpler. Okay, so keep that in mind, the partnership part of this. The Chesapeake Executive Council, which is up in that upper left-hand corner, are the governors who signed the watershed agreement. That's who's on that committee. Then it's very hierarchical, right? And there's a committee under them and then a committee under them. That's termed the leadership. The advisory committee is hooked to that leadership, right? What makes it important that this is a STAC report? As Brian alluded to, it's 38 experts from agencies, academia, private institutions, NGOs, and the watershed, right? So it's 38 people dedicating their time. It is herding cats, believe me, right? You all know what dealing with one academic is like. You ought to try like 38, getting them to all do the same thing. But STAC has an extraordinary amount of power in the Bay program. And I say it has 4 superpowers. One is that it's transdisciplinary. So there's somebody from every discipline. That means 2, it's able to synthesize. 3, it's independent. It gets to call balls and strikes. That's extraordinary, right? And it's consensus. It can provide a consensus view. So Kurt and I could be going all over the watershed, giving this spiel about this report, and it really wouldn't matter because we're 2 voices in the wilderness. But when it comes from STAC, that's a big deal. It's like the IPCC, right? It's a bunch of people willing to sign on and say, this is our best collective judgment. So I think that's why it's important. An advisory committee, if you ever get a chance to be on one, do it, because what's really interesting about it is there are lots of great scientists in the Chesapeake Bay program and in all these agencies, but they've got their heads down. And the STAC is the one body that gets to pick our heads up and go, you know what, you better pay attention to this, because this could really muck you up. And we get to do that. And so with that, I'm going to turn it over to my very capable colleague, Kurt.
I'm just going to put him on the stage. I'm going to stop sharing screen for just a moment, Kurt, so I can make sure that you're visible. So. Center. The stage, and now we're going to go ahead and share some slides again. So. This is where we were, right?
Yep.
Okay, so Kurt, just let me know when you want me to advance a slide for you.
Okay. Well, thanks. Thanks, Andy, for the invitation. And we have a lot to go through, so I won't dilly-dally. So I want to describe to you a little bit about what's in this report that we call the CESAR report. It's Comprehensive Evaluation of System Response to Efforts to Achieve the Water Quality Goals of the Chesapeake Bay. All I want to say is that we strove from the very beginning to involve all of STAC, all of that expertise that Denice mentioned before. And so it is presented as a report from the collective body. But with that said, next slide. But with that said, that pulling together this report sort of fell on the shoulders of the following people. So we had a steering committee that sort of guided this and guided the effort, overall effort. Those people are listed. And you'll also see the people listed in bold were a member of a smaller team that was primarily responsible for generating ideas, framing up the report, writing text. And those folks, what we call the writers group, they did the heavy lifting. So you see Zach obviously was a key member of that whole group from the get-go, as well as Leonard Shadman, Jeremy Testa, Denice, obviously, Mark Monaco, Kenny Rose, and Bill Dennison. So, just a shout out to those folks who had to endure. And the second thing I want to say is that this effort took 4 years, and we produced a lot of material. And this report is over 100 pages, but we didn't stop there. We had, we generated 3 other reports that were founded, that were, we call them STAC. They weren't consensus reports, but they were the product of a lot of expertise. And basically what we did is we gave people the opportunity to delve in the issues of CESAR in more detail, or to explore other issues, the related issues that necessarily weren't covered per se in CESAR, but were closely related, or to delve into a specific topic in more detail that you couldn't cover in CESAR. So we gave folks the opportunity to develop all of those things and to support this and to spin off of this overall CESAR document. So those are the documents that are listed. Those will be released when the report's released. There's a lot of great stuff in there. Next slide. So again, our focus, there's 10 goals of the Chesapeake Bay restoration effort. We're focusing on water quality. Just wanted to say that there's a lot of other things going on in the Bay program. This is where we're focused. Next slide. And the Chesapeake Bay program is celebrating its 40th anniversary today, this year. And if you go back and you look at the very first Chesapeake Bay agreement, the year it's all started, it was a 1-page agreement. And it basically said, it noted that there was a historic decline in living resources, and the major cause of that decline was water quality and poor water quality. And the chief culprit of that poor water quality was nutrients. That was a foundational goal, and it's still there. And it is the only goal of the Chesapeake Bay Agreement that has legal enforcement behind it, so legal requirements. So this is why we start that, and why we did this. 2025, we have a deadline in which the Bay program and the partners set for itself to try to achieve these water quality goals, and that's to have all pollution control measures in place to restore the bay, and to it's basically, and restore the bay means achieve the water quality goals that we established for ourselves. So next slide. What is CESAR about? This is a graphic that shows we have water quality goals throughout the bay, across different types of habitats and different type of constituents. And if you add those all up, and there's a percent attainment of those goals, and that's on the vertical axis. And the achievement of those goals are listed over time starting in 1985. And so you can see that there's a positive trend there. It's directionally correct. Not yet, but anyway, it's directionally correct. So we're having success, okay? A lot of success in the face of tremendous headwinds, right? Our population's growing, economic development's growing. We have more animals, more livestock, more agricultural development. Our climate's changing, and yet we were able to achieve this. But what we were looking at is why we are so far from the goal. Why are we sort of stuck at 30% achievement, it seems like? And after all, if you look, the Bay program, according to Bay program estimates, we've achieved our sediment reduction goals. We're getting close to achieving our phosphorus goals. Phosphorus tends to be a problem, but we can't overlook the fact that we've achieved 100 million pounds plus of reduction since 1985. Significant achievements. So why has this rate of progress been so slow? That's sort of the focus of CESAR. And so and then the second question is, what can we do to bend that curve? And what can we do as a program to improve the rate of effectiveness? Next slide. So just a quick background for the folks. This is the herding the cat, so to speak. This is sort of our approach of sort of framing up this problem and getting everybody sort of pulling in the same direction. Next slide. So across the top, this is sort of the policy sequence that we have to follow, that's being followed to achieve the water quality goals. The first step was back in the 2000s was to revise and refine our water quality standards. These set our goals. So if you hit the click, Denise, just to highlight this, the standards are sort of broken up in a couple of pieces. One is the ultimate use. What are you trying to achieve? And what we're trying to achieve, in the words of Kenny Rose, we're trying to enhance the critters in the bay, which are down in the bottom, the rock bass, the shad, so forth and so on. That is our goal, is living resources, to improve living resources in the bay. And how we do that, and how it was set up, is we defined conditions, water quality conditions, for instance, DO, that would be supportive of these different classes of species and organisms in the bay. And we defined those measurable criteria: dissolved oxygen, water clarity, chlorophyll A, across 5 different habitats. And you can see those in that diagram: deep channel, deep waters, open waters, shallow waters, and migratory fish and spawning habitats. So we set specific criteria, particularly dissolved oxygen and water clarity, across those 5 habitats to support their designated uses. The next thing we did, or next thing the program did, was say, okay, what stands in the way of achieving those criteria? Is nutrients and sediments loads. So the TMDL was a process to define what loads would be necessary in order to achieve those water quality criteria. And those are the loads that were estimated. And then we set off to define implementation programs to achieve the nutrient and phosphorus reductions that were necessary to achieve the criteria. Next slide. So what CESAR's doing is that there's a series of responses that have to occur in the watershed, in the estuary, in order to achieve these goals. And these sort of response functions are laid out below. And the report's sort of broken down and just looking at these response functions. So next, So we expect, for instance, implementation policies are intended to change people's behavior and change management actions in the watershed in order to reduce nutrient and sediment loads. We have an expected response. The Chesapeake Bay program is a sophisticated set of models to say, if we do this management action, we're going to get these set of reductions. And we think we know where we are on that response function. And that reductions are generating load reductions to the estuary. Then we look and say, we have another set of models to look and say, well, what if we given this load to the estuary, what's going to happen to DO, for instance, in these different habitats? And this is where we think we are, and where we're supposed to go is the blue star. Next slide. Now you'll notice that the living resource one is a little different. We have no expected response function here because the criteria were set up to achieve these dissolved oxygen criteria with the expectation that this would be supportive, and the program didn't really articulate, well, how would living resources respond to changes in water quality. So we expect that they're positive and have this type of a shape, and we can put on the vertical axis different measures of living resource response, abundance or diversity, whatever we want to put on that vertical axis, but that's really not articulated, but that's what we want. Next slide. So what we try to do in sort of framing up this question is try to go through what are some of the gaps and uncertainties that might be standing on our way, so to speak, to making more progress to achieving our goals. So let me just find a few terms that we use, concepts that we use. Forward, advance. We have something called an implementation gap, advance, which is basically the difference between where we are and where we want to go. Everybody's focused on that right now. Next slide. But we also know, everybody in this room also knows, that this is a very complex system. These are very complex response functions, and there's a lot of uncertainty around it in terms of where precisely we are, because you know, some models are useful, all models are wrong. So there is this uncertainty band around all these responses. Next slide. So given that uncertainty, there could be lots of different possible responses that might be different than what we are expecting to see. So one possible response, for instance, in the estuary and in the watershed, is that we might not be, for any given level of management actions, for instance, we might not be achieving the goals, the reductions that we think we are. In other words, the management actions may not be as effective as we think. Now, that white dot could be conceptually above the orange dot in terms of, you know, we could be getting more response than we thought. But all these, if you start looking at it, all the indicators are sort of suggesting, all the evidence is suggesting that we might not be on that response curve, and we might be lower than that. And so we're trying to figure out why that might be, where these response gaps, we call it, next slide, we call this difference of response gap. Where are they in the system? How much do we know about them? What can we do about them, if anything? Next slide. And then ultimately, some of the report that one of the things we wanted to focus on in the report is then to move beyond just the water quality criteria and say, what does it mean for living resources? Are there things where we do water quality improvement matters? What the conditions of living resources are that are not water quality related, like habitats and fishing pressure and harvest pressure, all of these other things also determine the water living resource response. So we wanted to look and say, what do we know about, how do we improve that living resource response, but we're not stuck in a low possible response, but we can have a higher possible response. And so one of the things that this group for the living resource group was looking at ways that we can better understand what kind of response that we'll be getting. Next slide. So how we broke this down into looking at these uncertainties and responses, if you think about the system as a whole, right, and we try to plot out the major sort of sequential actions that have to happen from policy intention to load reduction, to water quality response to living resources. And these sort of the major components. It looks complicated, but this is a very simplified diagram of the steps necessary you have to walk through. And so basically what we do is we're trying to break these down and say, what do we know about these? Where could these response gaps, where could these implementation gaps be in this long causal chain here? And slide. And I think one of the things was we wanted to stress is that, whoops, go back, and is that we wanted to stress and draw out the importance of behavior in this, because sometimes we forget about that. When we have implementation's got to go through people before anything's done. And so we wanted to understand that as better as well. And then the other thing we also wanted to stress is the water quality assessment is 2/3 of the way through this response. So if we're looking at achieving our goals, we're looking at dissolved oxygen, chlorophyll A, and water clarity. If we stop there, we're missing the boat potentially on how we can improve. What we really after is that last box, living resources. Next slide. So, you know, in the interest of time, I'm going to give you the bottom line now, and then we can iterate how we arrive at these conclusions in the next 15, 20, 30 minutes. But the bottom line is that these are significant gaps and uncertainties. They're presenting a major challenge to achieving water quality goals and improving resource response. These are formidable challenges. I don't think has been fully processed. A lot of people have been working on this. A lot of people are aware of the different problems, but when you string them all together, there's some significant headwinds here. And, you know, I think in the past, we were in this phase where we had big sources with pipes, and we're really past that now. And it's a non-point source world with millions of people making decisions over a vast landscape, and we have a fundamentally altered system. It is going to take some time, and it's going to be a challenge. And this is a polite way of saying that we don't know, and it's highly uncertain where we're going to be able to achieve all of the goals that we set out to achieve at the beginning. And if we do achieve them, it is going to take a long time. So the second point where the second conclusion is we don't know, and it's highly uncertain whether you can achieve all your goals, but we're trying to reframe the discussion in the sense of what can we do to, so to speak, to bend the curve, to change the trajectory so we can make more progress in terms of pollutant reductions on the non-point source side, more progress in water quality improvements, and most importantly, more progress in achieving improved living resource outcomes. Next slide. So I'm going to pause and take a breath. So I'm going to talk a little bit about the watershed section. And this, you know, hats off. This is the work of Zach Easton and a lot of stuff that happened in the car, and him, you know, we're sort of fleshing out these ideas back and forth over fires in the car. So this is coming from those conversations. So this is where we are on the implementation gap. I'm going to focus on nitrogen here just to illustrate a few points. Starting in 1985, we're at 370 million pounds of being discharged in the Chesapeake Bay. We're up, according to the Chesapeake Bay cast model, we're at around 257 now. 75% of those reductions that we've achieved have come from point sources and atmospheric deposition and reduction of air sources. So the gap, so to speak, the implementation gap is this difference, and it's about 40 million to 50 million pounds. And I could go into the details why it's more than what's shown on this graph because of Conowingo and climate change. But it's about a 50 million pound gap. Next. And to show you the world that we're dealing with, in this new world, this is a non-point source issue. 75% of the remaining load is resting with agricultural and urban non-point sources. All right, point sources, we've got a 65 million pound reduction since 1985. They're a little less than 30 million now. And so this is, the bottom line, this is a non-point source problem we're dealing with now into the future. Next slide. We've got to get out of those orange and green and blue bars, we've got to get 50 million extra pounds of reduction. Next slide. And this is going to be challenging through the non-point sources. In the last decade plus, we've achieved less than about 300,000 pounds a year. And at that rate, that's 150 years before we can add up to 150 pounds. To 50 million pounds of reduction. It is a challenging problem. And just to give you some perspective, a couple of wastewater treatment plant upgrades in Northern Virginia accomplished about what was accomplished in a decade of non-point source reductions across the entire watershed. So that gives you a little bit of perspective about what a challenge this is presenting. Next slide.
Bert, I'm going to give you a quick time check.
Yeah, what are we at?
I think you probably want to do at most 5 more minutes.
I mean, if people are willing to hang around a little bit, we could go a little longer.
Oh, I got the reprieve. So the response gap, so that's the best case scenario. The worst case scenario is that we've got these response gaps that we think in the watershed, in the sense that we're not achieving the goals. What we're doing now may not be as effective as we think they are. And so I want to focus the attention on that left-hand graph. So the orange on the chop tank, these are loads in terms of phosphorus loads since over the last almost 40 years. The orange graph is what we expect. This is what our models are predicting to happen to phosphorus loads. So you see this decline, but the red is what the trend that we're observing at a monitoring station. So the phosphorus trends are trending upward, at least in the chop tank, and you see a lot of watersheds like this. And the difference is this. Hit the slide. It's the response gap. That's what we mean by the response gap. It's the difference between what we're observing and what we're achieving. Next slide. And we see this, the monitoring data is a lot of evidence that shows that there's a lot of mixed signals about the effectiveness of agricultural and urban BMPs. And so the top graph is the Chesapeake Bay cast model and what they're predicting loads to be from 1982, 92, 2002, and 2012. And you can see that they're declining and for phosphorus and for nitrogen. But if you look on the bottom graph, this is the Sparrow model. And the Sparrow model is a statistical model looking at trying to explain trends across the watershed. And you can see, particularly in phosphorus, the trends are going in the other way. And you can also see, if you look at the nitrogen side, how sort of stuck we are on the non-point source side, which are the first couple of bars on those graphs. So next slide. Look, next slide. I'm not getting into this, but there's implementation gaps, and we've looked at what are behavioral response to the set of practices, set of programs and policies we have now for non-point sources. And if you look at the different types of practices you can do on the landscape, the things that people tend to adopt are things that have low upfront costs, we have high adoptions, and that have some private benefits. So that would be an example of cover crops. You're improving soil conditions. It's great for the farm. It's low upfront cost. But on the other end, a suite of BMPs, you've got expensive BMPs that have high installation costs, very few private benefits, but they can generate significant reductions, fairly certain reductions, and they may be a good deal on a cost per pound basis. They're very hard to get adopted. Next slide. I always complain about uncertainty, but Zach will tell me over and over again, there's one thing that he is certain of. If you get the mass balance right, you can make progress. And this shows the mass balance situation in the watershed. The left-hand side graph is for phosphorus, and this shows sort of loads where the import of the phosphorus may exceed what we can apply, what could be used for the land. And you can see those purple areas where there are mass imbalances. And if you go to the right-hand graph, the right-hand figure, you can see this is USGS's estimates of where the phosphorus loads are coming from, and the darker areas are higher loads. It's no surprise that, that those are the same areas. And those are also areas associated with intensive livestock production. So this mass balance issues are regional. And they are closely tied to intensive agriculture, particularly animal agriculture. Next slide. Like, I'm going to wrap up quick, but we're trying to figure out where these response gaps, what explains why we have these gaps across the landscape. There's a lot of legacy issues that could be masking the effectiveness of BMPs. We have uncertainties about BMP effectiveness, and there's all kinds of behavioral issues. Let's go next slide. This is just a graph of the legacy issues in the watershed. This is a graph of nitrogen concentrations in groundwater that's stored across the landscape, and anything in red is generally high probability of groundwater concentrations being above 3 milligrams per liter. Zach and I are working on a spring, and for instance, in the Shenandoah Valley has 6 milligrams per liter. It's twice the level of wastewater treatment effluent in most of the watershed. It's significant, and it's going to take decades for these to get out. And phosphorus, it's primarily phosphorus, excess phosphorus stored in soils. Next slide. It's going to take decades for this to work through the system, even if you remove applications now. We talk about BMP effectiveness. Are BMPs effective as we think they are? There's lots of reasons to think that they might not be. There's a lot of uncertainty around these. We're trying to generalize over complex situations. Next slide. And the last thing we're looking at is behavior on the landscape is very complex. It's difficult to observe, and it may not be matching what we assume. And I could go on and on about, not cover crops, conservation tillage. We're seeing some contradictory evidence in certain types of pollutants and certain types of situations where it's actually maybe making the problem worse. We have issues of differential adoption, like we're basing on average conditions, but we see behavior follows a distribution, and the tails might be really important in driving loads, but we don't know what those tails look like on that distribution. And model inputs, monitoring may not be sufficient. We may be making progress. Maybe our monitoring system's not able to detect a signal. Next slide. And the findings. Look, we think the bottom line is our non-point source programs right now, we think are insufficient to drive where you want to go. It's not just a money problem. We need to, and it's a tough nut to crack. Everybody's sort of facing this around the world and trying to solve, to reduce loads on a working landscape. It's tough duty. But we think if you're going to make progress, we're going to have to try some different things. And this next slide, and these are some of the different things that we're looking at and we describe in the report in terms of we got to shift focus and trying to focus more on outcomes and just implementing practices. There's opportunities for targeting investments. Loads are very unequally distributed around the landscape and across producers and across land managers. How can we better target our investments? How can we better incentivize people to target those investments? We need to experiment with different ways of doing things. We need to give an opportunity for people to try these different things. So we're suggesting things like sandboxing, where you give people an opportunity to deviate from the main rules and the main sort of frameworks. And to pick up on Zach's issue, focus on mass balance. It's the surest way that you can get to getting reductions. And a greater attention to uncertainty. There's a lot of things we don't know, and the program is really not built on trying to figure out these uncertainties. The program is sort of built on giving you specific answers, specific questions. So we need to focus more on trying to sort of parse out where those gaps are and how do we address them in the most effective way. I'm done.
Okay, all right. So Andy, I don't know if you need to do anything. I can.
I think you're being, people in the room can't see you on the screen, but they can see you in the room.
Oh, okay.
But I think you're fine. Just go ahead and talk.
All right.
You're visible on.
And so I'm cleanup batter. And so I'm going to talk about from here to the end. So you've got loads coming out of the watershed, and Kurt's talked to you about those. We're not seeing the load reduction we'd like to see. I'm going to talk to you about what happens once those load reductions get to the estuary and then all the way to living resources.
Oh, You got the slide, you can pass the slide.
Oh, from here. I
Getting
think
my
it's
phone.
working.
Is it frozen? Oh. Hang on. Did my screen just freeze up?
No, there's a menu open.
Oh. I don't know what it's doing. Hang on. Yeah, but I can't control the mouse. Hold on. Here we go.
Ah,
Now it should.
okay. So again, the questions are the same. What did we think was going to happen? And what has happened, or what evidence do we have to say what was the result? And if there's a gap, there's a response gap, how can we learn from that? So as Kurt, I'm not going to go over this a lot, but as Kurt said, we've made progress and nutrient loads have changed over time. So nutrient loads are about 15% lower from monitoring data than they were historically. And so you can see, you know, what really happened once the program began. So loads are going down by anybody's measurement. Loads are going down. And just to refresh you, if you don't know this already, but this is our most basic model of how the problem arose, right? So high nutrient inputs, you get high algal biomass, right? That you get turbid water from that, which means that the SAVs, the submerged aquatic vegetation, doesn't have enough light. Once those algae are sort of done, they die, they go down to the bottom, you get all this bacterial respiration that sucks all the O2 out of the bottom water, right? So high nutrient inputs, that's the chain of events. And the water quality standards are based on pieces of that puzzle. That's why there's a dissolved oxygen, a water clarity SAV goal, and a chlorophyll A goal, right? So they're measuring parts of that response chain. So the first thing you look at when you're looking at this chain of events is, okay, the loads are going down. Our total nitrogen and total phosphorus concentrations, there's 130 monitoring stations across the bay. It's a great data set if you need a project, right? And are those concentrations decreasing? And they are. 82% of those stations show lower nitrogen, total nitrogen concentrations over the period of load reduction. So good, check that box, right? So diminished loads, lower nitrogen and phosphorus concentrations. And if you're kind of saying, well, how does that work out spatially? If loads are going down, are concentrations always going down? Is that relationship, can we take stock in that relationship? And Jeremy Testa did this great analysis where all you really have to look at on this graph, I'm a walker and a talker. I usually have to take off a shoe to stay anchored here, so this is hard for me.
You're a camera no matter where you are.
Oh, well, okay. I want to point though, so if I'll do it this way. These are basically just a quadrant graph, and he's got total nitrogen load, the percent change on down here, and the total nitrogen concentration in the estuary change up here. All you really need to know are one of three things. All the green dots means loads went down, concentrations went down. Great. If it's red, load went up, nutrients concentrations went up. Check. The gray ones are where it doesn't make any sense at all. So these are for, the bay is split up into what's called 92 segments so that we can monitor them. 20 out of those 92, something different is going on. And so we can't bank on that relationship that, oh, okay, load went down, nitrogen and phosphorus will go down. And then if we take the next step and we go, well, really, we're reducing those nutrients to get a response in dissolved oxygen. And if you look at this map, this is the DO response. So we had nitrogen and phosphorus going down at 82% of the stations, right? At 82% of the stations, the bottom DO is either degrading or there's been no change. And that's what you see. If DO was going down and responding, there would have been a blue downward facing triangle. So you get two things out of this. One is the majority of the stations are not seeing a response in DO, but it's very spatially variable, right? There's really no spatial trend in there. Well, and I think this is a repeat of what Kurt started off with. If you look at overall water quality standards attainment, we seem sort of stuck between 30 and 40%. Well, what might be that be? And if you're looking at the Bay, are there ecologists? Right? The Bay is not one type of ecosystem. It's really a mosaic of about 5 types. We showed those at the beginning. Anything from shallow water to migratory and spawning habitat, open water, deep water, and then deep channel. What's on the bottom, that big channel that runs down through the main stem of the Bay. And so what you see is in migratory and spawning habitat, we have high attainment, but it's going in the wrong direction. And in the open water, it's kind of all over the board. And then in the deep channel, in red, it's low attainments, but it's headed in the right direction. So what are we to make out of all that? So this is the most, if I can explain any graph in the estuary section, this would be it. And so let me take some time to do that. The orange, first of all, you'll see a panel of 3, open water on the left, deep water in the middle, deep channel on the right. Percent attainment of the DO standard is on the y-axis. And what's total I'm just showing the results for nitrogen. Total nitrogen load is on the x-axis, right? So high loads are over on the right, low loads are over on the left. The orange circles are how partners set their plans for their management actions. They were called watershed implementation plans. The orange dots are the best estimate of what we thought would happen. So we thought that, for example, in open water, when we would get down to about a nitrogen load of about 200 million pounds per year, we'd be pretty much at 100% attainment. The blue dots are 3-year running averages from the time period 1985 to 2020. And they're the result of monitored results. Now, weather changes, precipitation changes, of course, that's going to be all over the board. There's going to be blue dots, you know, up and down that load spectrum. But what this says is, see where that red arrow is? That's a response gap. At low loads, we thought we would be up here in attainment, but the monitoring data says we're down here. There's 4 things I want you to take away from this series of graphs. And it has a lot of policy implications. The first is there's a response gap. There's a potential response gap. There's a lot of caveats about this data. This is trying to answer the question. But there seems to be a response gap in all 3 of those habitat areas. It's much larger at the lower loads. We're not seeing the response at the lower loads that we thought. That's where the response gap is bigger. And that response gap is the largest for the deep channel. So it's sort of saying the deep channel seems to be resistant to what we're doing in terms of load reduction. We are, however, I'll make a caveat and we'll say there have been, now that's Bay-wide, right? There is a response at local scales, no doubt. There's lots of examples. One of them is the Back River, where you see, and they're usually associated with point sources, wastewater treatment plants, and you'll get great improvement right in that local area. So why do we have response gaps? And I'm just going to concentrate on 2 factors that I think are having a lot to do with it. One is climate change. So there's a great paper, Frankel et al., just last year, that looked at, we're in the position now of saying, with climate change, well, what would have happened if we wouldn't have been doing load reductions? And you can see basically what this paper did was a great analysis and said hypoxia, right? Where we have less than 3 milligrams per liter of DO would have been much, much larger if we hadn't done anything about load reduction. And the bottom line is that they estimate that climate change has reduced the impact we get from reducing nitrogen loads by about 6 to 34%. So these temperature increases are doing things to the system that make it much, much harder to get a response out of our load reductions. The other one is an important concept in restoration in general. And so just follow the logic of this. If nutrient loads, increasing nutrient loads are on the x-axis, and hypoxia intensity, the area that doesn't have enough oxygen, is on the y. Degradation, follow the orange line, right? As loads increase, basically you start to get more and more hypoxia. And that green, this one, is sort of where you started to fall short of attainment, right? But when the system goes to recover, because of some feedbacks in the system can't go back down that same curve. The system goes back on the blue curve. And we can talk about why that is, but basically what it means is that then you have to decrease nutrient loads to a much, much lower, you're way, way left on the x-axis to get back to that attainment of water quality standards. Does that make sense? Okay, great. All right, so that happens in lots and lots of systems. Let me skip that. So there's all kinds of evidence in the Chesapeake Bay, not at the scale of the bay, but what we call subsystems, like in Mattawoman Creek is on the right, where we see these different trajectories where we can take advantage, right, of oysters or getting the water clear enough for the SAV, right? The system begins to accelerate its recovery, but there's often lag times. And so you can see in the Mattawoman Creek where you get actual, when you see a big reduction in about 2000 is when you got a wastewater treatment plant upgrade. And all of a sudden chlorophyll A went way down, right? Not as much algal biomass because there were less nutrients. And you can see there was sort of a lag time. The red line is water clarity. Took about 4 years, 4 to 8 years for the water clarity to respond. Once the water clarity responded, then the SAVs took off. And now the system is operating under a different set of rules. It's much easier. So basically, and then let me get to living resources and I'll try to be short. We have initiated a recovery, but the water quality response is less than we expected. And that's been especially slow in the deep channel. Why that matters is that many of the management actions and the policies that have been created in the Chesapeake Bay restoration are targeting the O2 in the deep channel, right? So if you're doing all this stuff that targets the deep channel and it's really intransigent, it's really hard to get there. Okay, so let me go through the living resources before I really tell you the implications of this. So we were just trying to answer the question, to what extent have living resources improved? And to go through this graph again, we don't have an expected response. Why is that? It's because the point of the Chesapeake Bay restoration was conditions for aquatic life. It didn't say make a calculation for crabs or oysters specifically. It said, let's make things better for this whole community of living resources. So there's not an expected response line. There are possibilities though. There's one that says, oh, we get a little bit of improvement from living resources with water quality attainment. And there's a possibility that there's a higher one that says we get more for those same water quality benefits. And there's, you know, a little bit to the shape of these curves, but maybe we can talk about that when we get to the end. This is a really important point. I don't know, none of you are old enough to kind of remember when we had audio receivers and we had a tuning knob to get the FM station. There was a really big knob for coarse tuning, and then there was this tiny little knob in the middle for fine tuning. And you really couldn't get fine tuning to cover large ranges, right? That's what we manage in the Bay is in red, DO, water clarity, and chlorophyll. We're on the, for living resources, we're on the fine tuning knob. There are things like fishing pressure, presence of wetlands, SAV, all these habitat conditions that have a whole lot more to do with living resources than this fine tuning button in the middle, right? And we have to keep that in mind. The other thing is that it's complicated. This is reinterpreted from a great paper that basically looked at sensitivities of different life stages of 4 different organisms to temperature, salinity, and DO. And all you have to notice from this is temperature is on the left, right? The order of their sensitivities is changing. So for example, if you're looking at a bluefish, you know, it's the most sensitive to temperature when it's adult, but when it's a juvenile, not so much. It's more sensitive to something like DO. So many knobs of living resource response. One of the things, or a stunning thing that came out of CSER is that, remember I told you, okay, we're chasing DO in the deep channel. Where does everything live? Where does most of the living resources live in the Bay? It lives in the shallow water. And oh, by the way, that's where people live, and most of the stakeholders really care about it. So one of the things is that if we were chasing DO in shallow water and habitat conditions in shallow water, that might really boost the living resource response instead of chasing the deep channel. Perhaps. And water quality alone does not improve living resources. As an example, there's a wonderful paper by Nick Licht and Secor, and it was looking at Atlantic sturgeon. And it said, if you met all the DO requirements in the Chesapeake Bay, you'd increase their habitat by 13%. If you raise water temperature 1 degree C, you diminish it by 65%. So that gives you a sense of the headwinds, as Kurt calls them. So there are opportunities basically to shift us to an upper living resource curve. So thanks for your patience. I'll just end with what Kurt started out with, which is achieving our desired incomes is proving more challenging than we thought. But there are opportunities. There is not a program in the country that has as much learning as the Chesapeake Bay does. And we need to take advantage of that. There's significant opportunities to improve our effectiveness, but in an old program that's hierarchical under the conditions of the TMDL, it's pretty rigid and it's going to be hard. This is something to sort of give you a thought exercise. The top graph looks at attainment of water quality standards on the x-axis and the cost to attain them. And a point I want to make is, and Kurt made this many times, you know, when you're getting wastewater treatment plant improvement, man, you're screaming across the bottom. You're getting lots of attainment of water quality standards for not a lot of cost. This is every program in the world. When it hits that non-point source issue, it goes, and all of a sudden the curve goes up and the implementation costs start to get great. And what this curve is suggesting is that it's going to get really costly if on the attainment, the deep channel is the last to come along. It's going to get really expensive to get there. And in fact, the cost may be more than we're willing to bear. Okay? So What can we do to move that cost curve to get more benefits for less cost? And by the way, in any sort of environmental restoration program, you're always making trade-offs. So when you're chasing DO in the deep trench, what are you not doing? Maybe what you're not doing is making sure you're installing living shorelines to boost living resource response, right? Or you're not paying attention to all the things you should, wetlands restoration. You're not paying attention to all those other things. The thing on the bottom says there are things you can do to make sure you boost living resource response for the same water quality management efforts. We know how to do that. And what can we do to make sure the program does that? So this is just a slide to say that this is the Clean Water Act policy process that Kurt had at the beginning. And you see it starts with water quality, the Chesapeake Bay Agreement, right, of which water quality is just the legally enforceable goal. The program has been down at the bottom of that, of implementation programs. How do we tweak BMPs? We're saying with Caesar, you have the opportunity to go back up into all those levels of decision-making and make changes. And how can we help them do that? And so that's it.
So let's thank our speakers first. Let's stop sharing screen and we'll take some questions. I know we're over time and I know a bunch of people probably have to leave.
I apologize, I can only talk
It's my
so
fault.
fast.
We started slow, so you know, that's just the name of the game. So what I want to do is to say, if there are people in the room who have questions, go ahead and we'll call on a couple. And then if people have questions online, we'll try to call on you as well. We'll take at least a few minutes to do this before we break. So
Yeah, let me just say, and I think I would express Kurt's feeling as well, that this is not our effort. This is the effort of like, what, 50, 60 people. And so we're honored to represent them.
So Denise, why don't you get back on camera? Anybody in the room have a question for either Denise or Kurt?
I don't have it well formulated yet, but something that we've been discussing a little bit around connecting science with policy and playing with the idea of when scientists should just be sharing their research and sharing that with policymakers for them to be developing policies, and when are people working in science like giving particular recommendations around those results. So I was thinking about that along with this report and thinking, is this, or I'm not familiar with how STAC usually shares guidance on next steps. Is it usually like BMP focused on which BMPs to be doing, or is it more like sharing the summary of what the research is showing to help guide the policy process?
Yeah, that's a great question. And Kurt can chime in. I can tell you for Caesar, we had to go through a, STAC can make recommendations. And we do science that informs policy. We don't make policy. That's clearly not our lane, right? But we can inform policy. And Caesar decided to go through, we have this interesting notion on the advisory committee, the advisory committee now, where we have representatives of federal agencies that bring their expertise to the advisory committee. And we've had to start this process where those agencies have to review our reports before their people can sign on as an author. USGS is the only federal agency without regulatory authority. I believe that's true. To pass USGS review, we cannot say you should do this. We can say, if these are your goals and you want to reach them in the shortest amount of time, you could do this. Does that make sense?
Yeah.
And the language is important, right? So we're informing policy, we're not making policy. And just as an aside, I mean, I think what's always been really wonderful about STAC's relationship to the program is that conversation between scientists and managers is really active. I mean, STAC, Andy knows as chair, when you're chair, it's often difficult to balance how much you're just responding to the program versus how much you get to have your head up and go, oh, this is on the horizon, you know? And so it's very responsive to the program, and that conversation is important.
That being said, this effort is the first time in how many years that STAC undertook something this large?
Yeah, it's been decades, and I can tell you that Kurt lost his hair and I went gray. We learned why you only do it once every 20 or 30 years, right? But I think that's important. It was important for us for it to be a consensus report, right? Because you just can't, it's hard to stand up alone and wave your hand in the wilderness.
Yeah, can I just add, I think that was a great question, and I just wanted to chime in and just reiterate what Denice said. You know, that's when we looked at that diagram at the beginning where you had the policy on the top and the response on the bottom. I think conceptually that sort of represents sort of our lane. We show trade-offs. We talk about the consequences of actions and the consequences of choices. But what you do with that information, that's above our pay grades. Like, you know, these are societal trade-offs and questions that somebody else has to decide. But, you know, but we can say if this happens, then this happens. And that's, I think that's our role, is to point that out as honestly as we can.
I would say too, just, you know, what's really missing in decision-making is, and this is in Kurt's lane more than mine, but is, you know, we're not very good at informing, well, decision-making under uncertainty is difficult. And where we really haven't gone, I think, far enough is informing decisions about trade-offs, because that's ultimately where you get, right? You can't, especially when you're on that part of the curve that I showed at the end, you know, you're going to start making trade-off decisions. And I wish we had more science basis to articulate those, if that makes sense. So for example, if you're, what does it mean to trade? What does it mean to maybe concentrate on doing things in the shallows to improve water quality there first to get an improved living resource response versus, you know, to keep doing things. This might be an aside, but you know, what controls DO in the deep channel is pretty much what happens up in the Susquehanna. So you can imagine there's this concept of most effective basins, right? Where the program gives resources to those basins up in the Susquehanna because they matter a lot to the DO in the deep channel. And one of the ideas in this report is, what if you reimagine those to include some shallow waters? What if you had most effective basins? Because these shallow waters are great test beds. That's where climate change impacts are happening first. That's where the living resources are, and it's where the stakeholders care about. So what if we sort of switch that thinking?
Or just a second, I didn't realize it was so focused on the deep channel and not much the shallow water. That's new for me to hear, so.
There's a historical reason for that, but it's probably would take too long to explain it.
Yeah, okay.
Okay, we are running late. Is there another question from the room, or is there a question online before we basically bring it to close? I'm not hearing any. So thanks very much, everybody, for attending.
Thank you.
For those, Molly, you want to say something? Maggie, sorry. Molly's over here.
Those who are joining for lunch will walk over to Sondheim Hall. We probably have a little more space than having an interest in having lunch. So you can join us. I think it has just been delivered by accident or by my mistake to the conference room in 210. And I know I said the cart lab. So depending on how many we are, we could just walk up to the conference room and he's there. If there are more clustering, then we can get down to the.
Okay, so thanks, everybody. Thanks so much, everybody online. Thanks especially Kurt and Brian and Zach. And A number of our colleagues are online as well. So I'm going to stop the recording now, and we will get this back and post it. We'll let you know when it's available for those who want to view.
I'll eat your lunch, Kurt.
Thank you.
All right.